Hello everyone, welcome back to your course on overview and integration of cellular metabolism. We are continuing with the carbohydrate metabolism. In the last lecture, we learnt about the various phases of hexose monophosphate shunt, and in this class we will be covering these following concepts. Okay. So, these two are covered in the last class, the hexose monophosphate shunt pathway and how it can be divided into, how it can be remembered easily by splitting the pathway into two phases, that is oxidative and non-oxidative phase. Okay. So, today we will be continuing with the regulation of HMP shunt, how the whole thing is regulated, the rate is controlled of the HMP shunt pathway, and also the metabolic and clinical significance of HMP shunt pathway. Why is it so important for us? Okay. So, this you already remember. Okay. From the last class, this is known as either pentose phosphate pathway or Warburg pathway or phosphogluconate pathway, and the importance was it is an alternative pathway to glycolysis and TCA cycle for oxidation of glucose, right. And I also told you the HMP shunt is actually more anabolic in nature. Why this is important? Because you already discussed in the regulation of enzyme class, knowing whether a pathway is anabolic or catabolic will help us to predict how it is regulated. Mind it, even though it is a pathway of oxidation of glucose, means ultimately the glucose molecule is broken down. However, the line is very important, read by the lines, it is more anabolic. There is a catabolic component, but it is more anabolic in nature because of the production of components like pentose and NADPH, which are further used to synthesize more components. So, this is the pathway which has been discussed in detail in the last class. So, if you cannot recall it right now, it is a very good time to pause this video, just recap the earlier class and then come back here again. Okay. So, the question arises in this class is how it is regulated. Okay. Means how the rate of HMP shunt is controlled. Very important. The one and only answer is by the cellular concentration of NADPH, the action of the enzyme glucose 6-phosphate dehydrogenase is controlled. See, this is the cycle, glucose 6-phosphate dehydrogenase was the first step, G6PD. The action of this enzyme is controlled. So, if we can control the action of this enzyme, the entire HMP shunt pathway, the flow will be controlled. And who can control this G6PD? That is NADPH or even NADP plus. It is the NADPH to NADP plus ratio. So, you can get any option. NADP is important, mind it is not NADH. NADH is very different from NADPH. We are looking for the P over here when we are dealing with HMP shunt. So, whether it is an important product or whether it is regulation, NADPH is your answer of choice. Okay. And what is the rate limiting step or the most important step with which we can control HMP shunt? That is glucose 6-phosphate dehydrogenase, either GPD or G6PD. This is the answer. Alright. So, since it is an anabolic process, now we extrapolate our concept that we learned in the very second lecture, that is regulation of enzyme action by covalent and hormonal modification. So, when a process is anabolic, what hormone in what form will help its progress? Anabolic insulin dephosphorylation, the rule applies here also. So, the synthesis of glucose 6-phosphate dehydrogenase, that is from glucose, is actually induced by the increased insulin to glucagon ratio, means more the insulin is, the more will be the pathway. When is the insulin level high? After a carbohydrate rich meal. So, when body is rich in carbohydrate, insulin level goes up, insulin helps in this pathway. Okay. So, insulin will induce glucose 6-phosphate dehydrogenase and overall increase the rate of the pathway. Okay. So, this and this was about the oxidative phase, because in oxidative phase glucose 6-phosphate dehydrogenase acts, right. And what about the non-oxidative phase? So, in oxidative phase who was getting produced? Mainly carbon dioxide, right. And in the non-oxidative phase various pentoses were produced. The non-oxidative phase is regulated by the requirement of pentoses. How much pentose is required for the body? Why pentose is required for the body? It is required for production of DNA and RNA. If we need pentose, we need more source of pentose, that is HMP shunt, and then also HMP shunt will be upregulated. When there is no requirement of pentoses, when there is less insulin and high glucagon, the process will be inhibited. It is as simple as that. Now, we already have discussed briefly the significance of HMP shunt, that is it plays a major role to produce two most important compound, number one pentoses or pentose sugar for formation of DNA and RNA, as well as NADPH. Okay. I also told you something in last class. It might appear trivial, but it's very important. That is 95% or more than 95% of the body's NADPH is produced by hexose monophosphate shunt, and less than 5% is produced by malic enzyme. Malic enzyme, not malate. Okay, malic enzyme. It's quite different. So, what is the importance of pentose? The hexose are converted to pentose. We already know how hexoses are rearranged to pentoses, and it is important in synthesis of DNA and RNA. Deoxyribonucleic acid and ribonucleic acid, the very fundamentals of life, the very fundamentals by which DNA and gene and chromosome are made of, right? DNA makes chromosomes after supercoiling. So, those ribonucleic acid are derived from ribose, which is a pentose produced from HMP shunt. Next, what is the importance of NADPH? NADPH has got a lot of functions in the body. Right way we can see, we will first enlist what are the functions, then we will delve into small details which is required for you. So, number one, it is required for biosynthesis of fatty acid and steroids. We will be discussing them later during the course when we are dealing with lipid metabolism. Apart from that, NADPH is used in synthesis of certain amino acid involving the enzyme GDH or glutamate dehydrogenase. We will cover in detail when we are dealing with protein and amino acid metabolism, but for now you should note down these examples so that you can recall later, because everything is linked, right? We will also tie up everything during integration of metabolism session. Anyway, next most important activity of NADPH is for a defense mechanism. I told you during the course outline we need to learn metabolism so that cell, why metabolism is important? Because cell needs metabolism to defend itself against external stimulation, external infection. This is the key area, because NADPH or nicotinamide adenine dinucleotide phosphate, the reduced form NADPH, is actually responsible for production of reactive oxygen species by macrophages which help in killing the bacteria. How does it happen? You see, the phenomena is known as respiratory burst. Okay. So, respiratory burst is the phenomena by which multiple compounds starting with NADPH are involved in production of reactive oxygen species or ROS, which ultimately acts as a defense mechanism to kill the bacteria. See how this occurs. We can get NADPH from glucose. This is the HMP shunt pathway, okay, by the enzyme glucose 6-phosphate dehydrogenase. On NADPH, the enzyme NADPH oxidase acts. So, when NADPH oxidase acts, it is reduced to NADP plus. This hydrogen, that is the nascent hydrogen, is used, or this electron, okay, reduction oxidation is an exchange of electron. This actually is used to convert oxygen to superoxide radical O2 minus dot. This is superoxide radical. Okay. So, NADPH oxidase enzyme is actually producing superoxide radical, which is further acted upon by the enzyme SOD or superoxide dismutase, full short form is SOD. So, SOD acts on O2 minus, that is superoxide radical, to produce H2O2 or hydrogen peroxide, right. Next, in next step what happens? Myeloperoxidase or MPO acts on this hydrogen peroxide in presence of chloride ion to produce hypochlorous acid. Hypochlorous acid, or H, it is also denoted by HOCl, HCl or HOCl. This hypochlorous acid as well as the superoxide radical and even in some cases the H2O2 can directly attack and damage the bacteria or the foreign invaders. Okay. So, it helps in production of reactive oxygen species, and it is the reactive oxygen species which are arrows by which a cell shoots the arrows at those foreign bacteria and they die, as simple as that. But you know what happens? There is a problem to the whole thing, because once the bacteria are gone, okay, arrows are shot, 100 arrows have been shot, 50 have killed the bacteria. So what will happen with the rest 50? If left just like that, if ROS or reactive oxygen species is just left roaming in the system, what will it do? It will attack our own RBC membrane. So these free radicals that are generated needs to be scavenged, needs to be taken care of. Who does this? So first of all, why it is important to scavenge the free radicals? Because as I told you, the free radicals are constantly produced inside the cell, and if left unchecked, they will destroy the DNA, protein, fatty acid, and all biomolecules will be destroyed. It will create a chain of cellular oxidation reaction. It will lead to lipid peroxidation, and the entire cell membrane will be damaged, cell membrane, DNA, RNA, everything. So this free radical is actually, there is a mechanism by which this free radical can be checked, the progress of this free radical, the activity of this free radical, so that they are not unbound to do anything they want. After the bacteria is killed, you need to come back home. Suppose this is our hitman, okay, in the cellular system. We have given a contract to kill the villains, that is the bacteria. So what they do? They are checked by multiple enzymes. The most important of that is glutathione peroxidase and glutathione reductase, also superoxide dismutase. So mind it, superoxide dismutase is a part of generation of reactive oxygen species. It also tackles some reactive oxygen species by scavenging them. But the most important is this word glutathione, and especially is in the reduced form. And who helps in reducing glutathione? The enzyme glutathione reductase. Now the question is, we will be seeing everything in a diagram very soon. Who actually helps in reducing glutathione? For reduction we need a hydrogen ion, right? This hydrogen ion is actually supplied by NADPH. So watch very carefully what is happening over here. Now we are trying to tackle the free radicals that are already in excess. So NADPH, by the action of NADPH oxidase, was generating free radical, one paradigm. Next is, so again we are getting NADPH from glucose via the HMP shunt. What is happening? The hydrogen from NADPH helps in maintaining glutathione in its reduced form. So this is the oxidized form of glutathione, and this is the reduced form of glutathione. This is denoted by GSH, two molecules of reduced glutathione, G2GSH, and when in oxidized form they actually dimerizes to form GSSG, a disulfide bond is there. So remember, anywhere you are listening to the word thio, it means sulfur, sulfur is there, okay. You might be knowing that from your chemistry knowledge already. So what happens? So these glutathione peroxidase is a variety of peroxidase. So what it will do? It will convert the peroxides to harmless water, right. So where from we are getting peroxide? Superoxide dismutase can convert all free radicals, the reactive oxygen species, that is O2 minus etc., to H2O2, okay. This you already saw in the previous mechanism. H2O2 after being produced, when we are killing bacteria, myeloperoxidase act on that. But here glutathione peroxidase is acting to produce water, which is absolutely harmless to the system, right. So what will happen if, so can you guess, if this thing is jeopardized, what will happen? See, there are multiple sources of free radicals. Free radicals are actually not only produced by NADPH oxidase, there are other multiple cellular sources of free radical which are produced from either cellular respiration at mitochondria, oxidation. There are oxidative drugs, there are multiple sources. However, this is the only source or major source by which all the free radicals can be scavenged. So if there is a deficiency of NADPH, okay, there will be less and less glutathione, reduced glutathione, there will be less and less scavenging of free radicals, and this free radical will ultimately destroy all the membranes, especially the RBC membrane, in which there is a huge amount of HMP shunt going on, okay. So this is an applied question right over, and how NADPH can be deficient? Can you tell me? Glucose produces NADPH by the action of glucose 6 phosphate dehydrogenase. We are talking about deficiency of NADPH. So, who is at fault? Definitely this. If this enzyme is deficient, there will be problem. We will be discussing it very soon. So, this all leads to an loss of integrity in RBC membrane. So, normally RBC membrane is maintained intact due to action of NADPH, glutathione and glutathione reductase that I have already explained in the previous diagram. So, if those free radicals are left unchecked, it will lead to loss of or destruction of RBC membrane. So, in when we are looking at the beneficial role, NADPH, glutathione and glutathione reductase actually helps to maintain the integrity of RBC membrane. Since NADPH, the major source is HMP shunt. So, the existence of HMP shunt is actually protecting the RBC or erythrocyte membrane from free radical damage. Okay. The next importance of NADPH, mind it, we are discussing the role of NADPH. Okay, I hope you are there with me. So, NADPH is also important in prevention of methemoglobinemia, right? What is methemoglobin? Methemoglobin is the oxidized form of hemoglobin. Remember, in hemoglobin, that is the pigment that is in RBC that is carrying oxygen, is in ferrous form, Fe2 plus. It has got a tendency to be oxidized to ferric form, Fe3 plus. If iron in hemoglobin is oxidized to ferric form, hemoglobin is converted to methemoglobin and it loses all capacity of carrying oxygen. Okay, methemoglobin cannot carry oxygen. And who helps to maintain the reduced state? Again, NADPH. So, NADPH is required to keep the iron in ferrous state and to prevent methemoglobinemia. So, again, if there is deficiency of NADPH, there will be methemoglobinemia, right. Next, NADPH, again one of the very important uses to help in detoxification of drugs. You just need to know most of the drugs that you are consuming, medicines, okay, they after their action they need to be rendered useless. It happens in liver in the microsomal oxidation system, and it uses an enzyme system that is known as cytochrome P450 based oxidation system. Cytochrome, it is an iron containing pigment. So, cytochrome P450 based system is helping in detoxification of drugs in liver. And who helps in this cytochrome P450 system? Again, NADPH. So, if there is no NADPH, there will be no detoxification of drug, or there will be very reduced detoxification of drug using cytochrome P450 microsomal enzyme system, and suddenly all the drugs will have more and more side effects. Okay. Next, NADPH is also important in eye to maintain the transparency of the lens. Okay. Just know this, NADPH is very important in preserving the transparency of the lens, because if there is excess oxidation of the products, that will lead to osmotic imbalance, there will be water intake and all the refractive index of the lens will be blurred, and then we will have something known as that will be very problem, vision will be blurred, right. So, NADPH being present in the lens also helps to keep the essential lens components in reduced form and thus maintain the transparency of the lens. So, what happens over here? So, for production of NADPH, we already learned the main important enzyme, the key enzyme is glucose 6-phosphate dehydrogenase. After knowing all these things that glucose 6-phosphate dehydrogenase is doing, and after all the beneficial roles of NADPH, you can easily guess what will happen if there is a deficiency of glucose 6-phosphate dehydrogenase. So, even if you do not watch this video along, you can pause this video and you can actually make your mind and just make few notes what will be the problem. Let the video run and you can tally later where you write in predicting. So, let us see. Okay, first of all, the G6PD deficiency is a sex linked trait. Okay. It means what? It is always dormant in females because there are 2 X chromosomes. It is an X linked recessive disorder, and only in male patients or male individuals where there is only one X chromosome and one Y chromosome, they can be expressed. So, all G6PD deficient cases are males, females are carriers of G6PD deficiency. Fine, mind it, males are patient and females are carriers. They are patients or their cases, right. So, decreased G6PD impairs the synthesis of NADPH in RBC, that will lead to destruction of RBC membrane and that will lead to hemolysis, right. This hemolysis is more and more pronounced when the body is all under some oxidative assault. For example, drugs like primaquine. There is an anti-malarial drug chloroquine and primaquine. Primaquine is very essential for treatment of malaria. For drugs like primaquine and few vegetables like fava beans, okay, they contain some products which leads to increased oxidative cellular environment, and when their oxidation is happening more and more, oxidative drugs, they produce more and more free radicals, and we need the action of NADPH, glutathione reductase, etc. to scavenge those free radicals. So, a normal person, you and me, who does not have any problem, can easily intake primaquine, fava beans, and we are fine, because all the NADPH are scavenging the free radicals. What if this NADPH is deficient because of deficiency of G6PD? Then suddenly, maybe normally we are going fine with some amount of NADPH that is being present, okay, from malic enzyme, but as soon as there is an oxidative assault, there will be profuse hemolysis, because there is no one to scavenge all the free radicals and they will destroy the RBC membrane, right. So, if there is an excess production of free radicals, so we need more and more reduced glutathione, we need more and more NADPH, but if this is deficient, there will be no NADPH, there will be no reduced glutathione, and ultimately these free radicals will destroy the RBC membrane. The membrane will be totally destroyed, and that will lead to a disease known as hemolysis, that is destruction of red blood corpuscles. Now, after this hemolysis occurs, what happens? The first of all hemoglobin goes out of the cell, and what happens? This hemoglobin can be no longer maintained in the reduced form, because I told you NADPH is also essential in preventing the conversion of hemoglobin to methemoglobin. So, there will also be methemoglobinemia. So, when observed on the microscope, these hemoglobin molecules cross-link with each other to form something known as Heinz body. Again, very important MCQ question. So, membranes are and these Heinz body also plays a role in damaging the RBC membrane. So, RBC membrane by Heinz bodies and reactive oxygen species due to deficiency of NADPH, which is due to the deficiency of glucose-6-phosphate dehydrogenase, leads to hemolytic anemia. All the RBCs will be gone, hemoglobin concentration will fall, anemia will happen, and it is a condition known as hemolytic anemia. Okay. So, G6PD deficiency and malaria. Okay. Now, G6PD deficiency is actually associated with resistance to malaria. This is a protective mechanism that is found in African population. So, what actually happened over here with adaptation, the malarial parasite that is invading the red blood cell actually needs reduced glutathione for survival, right. And the African population, some areas of African population has been seen they are born with glucose-6-phosphate dehydrogenase deficiency, right, and those areas are actually prone to tropical diseases like malaria. So, imagine the situation, what happened if they are contracting malaria, if they are having malaria, the disease, if they are giving primaquine, hemolysis or hemolytic anemia will occur and the patients will die. Naturally, I mean luckily, they have been protected by evolution in such a way that the malarial parasite itself needs reduced glutathione, right, to survive. So, in those patients where glucose-6-phosphate dehydrogenase deficiency is there is no NADPH and there is reduced glutathione is also less. So, the malarial parasite dies in the first place. So, they are naturally protected from malaria. So, this is an example of a natural adaptation where an enzyme deficiency in a specific geographical region is protecting the population from any disease, the disease being malaria. Next, thiamine deficiency. Did you remember the enzyme transketolase, was catalyzing transfer of carbon atoms from 5 carbon to 3 carbon to the formation of, you remember in HMP shunt. If you don't, please, there are 2 transketolase reaction. You should remember that. You should pause the video, look at the previous class, look at the pathway of HMP shunt and then come back here. Over there it was mentioned the thiamine is actually the cofactor of transketolase enzyme in the form of TPP or thiamine pyrophosphate. Okay. So, it is so much dependent that actually the deficiency of vitamin B1 or thiamine is checked by estimating the transketolase activity. So, if the transketolase activity is down, the vitamin B1 deficiency is there, and this is actually manifested by a disease that is known as Wernicke-Korsakoff syndrome. In many textbook the disease can be split. I mean, the milder phase is known as Wernicke's encephalopathy and the severe phase is known as Korsakoff's psychosis, but in many textbook they are clubbed together. So, Wernicke's or Korsakoff's, you mind that this is vitamin B1 deficiency thiamine testing by transketolase activity. What are the features? The features are loss of memory, mental disorder and partial paralysis. So, these are the significance of HMP shunt that have been discussed. So, reductive biosynthesis of fatty acid, cholesterol and steroid hormone. For next set of classes, free radical scavenging, maintaining the integrity of RBC membrane, prevention of methemoglobin formation, detoxification by hydroxylation, maintaining the transparency of lens, bactericidal activity of macrophages. This will be discussed later, okay, or even in live classes for that matter. So, to produce, and the importance of the non-oxidative phase is to produce DNA and RNA with the help of pentoses, and clinically what we need to remember is glucose 6-phosphate dehydrogenase deficiency that actually leads to so many problems. Okay. So, the whole problem is due to this G6P deficiency, and this also leads to drug induced hemolytic anemia, methemoglobinemia as well as thiamine deficiency that leads to reduced transketolase activity. So, the key points in this class were the whole combined 2 classes is NADPH is produced as well as pentoses are produced from HMP shunt. The first reaction, that is catalyzed by glucose 6-phosphate dehydrogenase, is the rate limiting step and it is inhibited by NADPH as an allosteric inhibitor. The oxidative phase is controlled by the level of NADPH and non-oxidative phase is controlled by requirement of pentoses, and insulin induced glucose 6-phosphate dehydrogenase and therefore increase the overall pathway. Okay. So, these are my references and thank you all for your attention.